Dear students, welcome to the organic chemistry topics of semester 6. In this lecture, we shall discuss about proteins. Proteins are macromolecules. Their molecular masses range from few thousand to several million. The building blocks of proteins are alpha amino acids. Thus, proteins are the polymers of alpha amino acids. A total of 20 different amino acids exist in proteins, and they are arranged in different sequential orders. Proteins differ from each other primarily in their sequence of amino acids. Amino acids are released from proteins by hydrolysis. Proteins perform a vast array of functions within the organisms, which include metabolic reactions, DNA replication, structure of cells, transporting molecules, etc. The structure of an amino acid is represented here. Proteins are polymers of alpha amino acids. In alpha amino acids, the amine group is attached to alpha carbon. That is, this is the alpha carbon. The carbon next to the carboxylic acid group is called the alpha carbon. The amine group NH2 group is attached to the alpha carbon. An R group is attached to the carbon, also a hydrogen, and thus the 4 valencies of carbon are satisfied. The simplest amino acid is glycine. It is otherwise called 2-aminoacetic acid. In glycine, the R group is the simplest hydrogen atom. So, the 2 valencies of carbon are satisfied by hydrogens, one by carboxylic acid group and other by the amino group. Next is alanine, in which the R group is CH3. Alanine is 2-aminopropanoic acid. Another amino acid is valine. It is 2-amino-3-methyl butanoic acid. The R group is CH3 twice CH. Except glycine, all amino acids are optically active because all amino acids contain a chiral carbon. In the case of glycine, there is no chiral carbon because the alpha carbon is attached to 2 hydrogens. Thus, except glycine, all amino acids are optically active. There are different types of classifications for amino acids: structural classification, electrochemical classification, and biological classification. In structural classification, amino acids are classified into acyclic and cyclic. Acyclic amino acids are again classified into different classes. First is aliphatic unsubstituted amino acid. Example, glycine. Next is hydroxy amino acid. The amino acid contains an OH group. Example is serine. thioamino acid. It contains an SH group. Example cysteine. Carboxy amino acid. Example is aspartic acid. The amino acid contains an extra carboxylic acid group. Diamino acid. The amino acid contains an extra amine group. Example lysine. Cyclic amino acids are classified into aromatic amino acid. Example is phenylalanine. Heterocyclic amino acid, in which there is an heterocyclic ring. Example tryptophan. And the last one is rare amino acid. Hydroxyproline is an example for rare amino acid. In biological classification, amino acids are classified into essential and non-essential amino acids. Essential amino acids are those which cannot be synthesized by human body from other compounds, and hence these amino acids must be supplied in the diet. Example valine, leucine, isoleucine, tryptophan, etc. Non-essential amino acids can be synthesized by human body in sufficient amounts. Examples include glycine, proline, aspartic acid, etc. In electrochemical classification, amino acids are classified into acidic, basic, and neutral amino acids. As the name indicates, acidic amino acids have enhanced acidic character due to the presence of an additional carboxyl group in the side chain, in the R group, R side chain. Basic amino acids have enhanced basic character due to the presence of an extra amine group. In neutral amino acids, it contains equal number of acidic and basic groups. Aspartic acid is an example for acidic amino acid. In aspartic acid, the R group is CH2COOH, so that the amino acid has an extra COOH group. Lysine is an example for basic amino acid. It has an extra NH2 group. The R group of lysine is CH2 4 times NH2. Since it contains an extra NH2 group, lysine is basic in nature. Glycine, alanine, etc. are examples for neutral amino acids which contain one COOH group and one NH2 group in their structure. Due to the presence of an acidic and basic group in the same molecule, alpha amino acids exist in an ionic form. This ionic form is called Switter ion or dipolar ion. In the Switter ion or dipolar ion, the hydrogen of the COOH group is transferred to the NH2 group. When the hydrogen is transferred to NH2 group, nitrogen gains a positive charge and there exists a negative charge on the carboxylic oxygen. A sweeter ion or a dipolar ion is an internal salt formed by the interaction between the acidic and basic groups that are the part of the same molecule. The amino acid sweeter ion can either donate or accept proton depending on the pH of the medium. At low pH or in acidic medium, the sweeter ion accepts a proton and becomes a positively charged ion. The accepted proton gets attached to the oxygen of the carboxylic group. In basic medium or medium with high pH, in the presence of OH minus, the sweeter ion donates a proton and becomes an anion. So for each amino acid, there is an intermediate pH at which the amino acid will exist as a sweeter ion and carries no net charge. The positive charge on nitrogen is cancelled by the negative charge on carboxylic acid group. And this particular pH for an amino acid is defined as the isoelectric point of that amino acid. The pH at which an amino acid sweeter ion carries no net charge and hence shows no tendency to migrate either towards the cathode or to the anode when placed in an electric field is called the isoelectric point of that amino acid. If the amino acid is positively charged, it has a tendency to migrate towards the negative electron. If it is negatively charged, it has a tendency to migrate towards the positive electron. At a particular pH, the amino acid will exist as the sweeter ion with no net charge and hence when placed in an electric field at this pH, the amino acid will not migrate to any of the electrons. And this particular value of pH is known as the isoelectric point of the amino acid. The isoelectric points of neutral amino acids will be close to 7. That of the acidic amino acids will be less than 7. It will be around 2 and 3. And for basic amino acids, the isoelectric point will be above 7.